replication is a process that allows for the inheritance of genetic information. DNA replication takes place in the nucleus for eukaryotic cells and the cytoplasm for prokaryotes. An enzyme called helicase separates the two parental strands of DNA by breaking hydrogen bonds holding base pairs. Each of the separated parental strands serves as a template for the synthesis of a daughter strand. Topoisomerase works upstream from the replication fork to relieve the stress that results in the unwinding completed by helicase. DNA nucleotides consist of a 5-carbon sugar, deoxyribose, the 3 phosphates, and a nitrogenous base. These are adenine, thymine, guanine, and cytosine. RNA primase builds an initial string of RNA that is complementary to DNA in order to initiate DNA replication. Both the lagging strand and the leading strand begin with a stretch of RNA called a primer. Once the primer is laid down, DNA polymerase adds successive nucleotides to the daughter strand, using the other as a template. The leading strand gains nucleotides in the direction of the replication fork, but the lagging strand is synthesized in pieces. RNA primers are laid down periodically, and its 3' end is pointed away from the replication fork. This causes Okazaki fragments. After synthesis, DNA ligase removes RNA primers and connects fragments. The 3' OH of the last nucleotide on the new strand attacks the 5' phosphate of the incoming nucleotide, resulting in 2 phosphate groups. The sugar and phosphate form phosphodiester bonds, establishing the backbone in DNA. DNA is altered when DNA polymerase fails to proofread the bases. On rare occasions, an incorrect nucleotide is attached to a new DNA strand. Base substitutions, deletions, or insertions may occur. When base substitutions occur, replication can be affected by one nucleotide swapping for another, causing an incorrect base pairing. When deletions occur, one or more nucleotides are removed and the genetic code is read by RNA, which can cause defective proteins. When an insertion occurs, an extra nucleotide or more is added, disrupting the amino acid sequence. These changes can alter the natural shape and therefore function of proteins. During transcription, which takes place in the nucleus for eukaryotes and cytoplasm for prokaryotes, RNA is synthesized with a template strand from DNA. A nucleotide RNA is made of ribose, 3 phosphate groups, and a nitrogenous base. In RNA, thymine is substituted for uracil. Helicase also unwinds a double-stranded molecule of DNA, and transcription factors, proteins that bind to the DNA molecule to begin transcription, assemble at promoter regions along the DNA. Promoters are regions of DNA that initiate genes. RNA polymerase is put into place between the strands of the DNA double helix, and the transcription initiation complex is activated to add nucleotides to 3' end of the transcript. RNA polymerase unzips a small portion of the DNA helix, and one strand acts as a template for the synthesis of an RNA molecule, which is synthesized with RNA polymerase. RNA is synthesized in a 5' to 3' direction, anti-parallel to a template strand. Once RNA polymerase reaches the terminator region, the process stops, and then RNA is released from the nucleus through nuclear pores in the cytoplasm. In eukaryotes, transcription results in a primary transcript that can be modified to become mRNA. In prokaryotes, transcription immediately results in mRNA.
Finally, translation takes place in the cytoplasm for both eukaryotes and prokaryotes. More specifically, it takes place on ribosomes that synthesize proteins. Ribosomes are composed of a small and large subunit which are made of rRNA and proteins. Translation begins with the translation initiation, where the start codon is recognized and a polypeptide begins. Initiation factors bind the small subunit mRNA and a tRNA containing methionine. The initiation complex of translation moves along mRNA until it reaches the start codon. When it reaches AUG, initiation factors are released and the large ribosomal subunit joins the complex. Then rRNA works to read incoming codons and link amino acids together. Codons are groups of 3 adjacent nucleotides in mRNA that specify one amino acid in a protein. The position of a start codon establishes the reading frame that determines how mRNA is read. In translation elongation, amino acids are added to the growing polypeptide chain. tRNA is attached to a specific amino acid as well as an anticodon that consists of 3 nucleotides that undergo a base pairing with the opposite codons in mRNA. tRNA brings the correct amino acid to the ribosome, and once it's bound to the ribosome at one site, the next tRNA lines up to bind at the site adjacent to the first. Then a dehydration synthesis reaction occurs in order to form peptide bonds. The ribosome shifts codons, and downstream codons are read. The amino acid end on tRNA containing the anticodon attaches to the chain of a polypeptide, becoming the new carboxyl end. This process repeats until termination, which takes place because a stop codon is in the mRNA is met by the ribosome. A protein release factor also binds the ribosome, causing the bond between the polypeptide and tRNA to break, creating the carboxyl terminus of the polypeptide and the large and small subunits to dissociate from each other once the polypeptide is released. Thank you.